Hi, I'm Charles Yanofsky. I'm Professor Emeritus at Stanford University.
I'm David Botstein. I'm still a professor at Stanford University. When is the first time that you remember thinking about being a scientist?
Probably when I was considering what high school to go to.
Mm-hmm.
I grew up in the Bronx. I enjoyed nature, what little there was of it outside of apartment houses in the Bronx. I used to chase butterflies. I lived next to New York University campus, and they had wonderful fields, and the monarchs used to come there every summer, and I loved chasing monarchs. So I was interested in nature, and I also had given a chemistry set as a child, like most of us were, and I enjoyed making explosives for chemistry, build rocket ships with explosives, but I became fascinated by chemistry, visited chemistry shops and looked at what they had to say. And also, my mother thought it was important for her kids to escape from the city in the summer. So me and my sister and brother used to go to summer camps every summer. Mm-hmm. And in summer camps we'd see snakes and wild animals, and I was fascinated by this. But I think probably the major factor that led me to consider going to a science high school was the kids I grew up with. The boys and girls in my neighborhood were interested in everything that was going on in the world, and were interested in nature and everything else. And quite a few of us went to the Bronx High School of Science. And you know, too, the Bronx High School of Science, the teachers there were just unbelievable.
I guess it's worth explaining that we both went to the same high school,
Yes.
Nearly 20 years apart.
The faculty, teachers at the Bronx High School of Science who just made a point of telling us how nothing could be more exciting than trying to figure out what goes on in nature, whether it's chemistry, physics, or biology. They just kept making that point, and almost everyone was convinced. It seemed that everyone graduating Bronx High School of Science wanted to become a science major when they went to college. I was primarily interested in chemistry, not biology, when I went to high school. And but I found genetics fascinating, and we had something called Drosophila Squad at the Bronx High School of Science. We played with Drosophila. And I actually stained some salivary chromosomes, and I thought that was fabulous. Uh-huh. So we went on outings to interesting places, and we went to the Cold Spring Harbor Laboratory and heard lectures by prominent geneticists. Someplace along the line, I was convinced that you had to apply biochemistry to answer the questions of genetics and biology. I was convinced. So I was mostly interested in biochemistry in high school.
So this is before DNA.
Way before DNA.
So the idea that biochemistry was related to genetics was not self-evident at that time.
No, I didn't even, in fact, didn't learn about it until after World War II, when I went to see the chairman of the biochemistry department at CCNY and told him I had a problem. I was interested in both genetics and biochemistry, and so what do you do? I didn't want to go to graduate school and major in one or the other, and he took down his copy of Benjamin Harrow's textbook, and there was one paragraph on betel and tatum and the one gene, one enzyme hypothesis. And I read that, and that was it. I was hooked from then on.
You had to interrupt your college studies in World War II.
Yes, so I enrolled at CCNY as a biochemistry major, and after a year and a half, I reached the age when I was drafted. This was 1944. They gave us intelligence tests and whatnot. So they picked out about 80 of us from the New York area, and they put us on a train. We didn't know where. We ended up at Fort Blanding, Florida. The only thing we could find out that we had in common, we all had some time in college. And we were assigned to a cannon company, infantry cannon company. And we found out that the reason that the 80 of us were picked to go together is that there were something like about 300 recruits trained in the same division, same group, and the others were being taught to load shells into cannons and to do all the hard work, and we were taught how to aim the cannon. So the thought was that we were college students, we could learn how to aim a cannon and fire it, and this was in July of '44, just after the invasion in Europe, and most everyone there was sent to Europe to replace killed or badly wounded American soldiers, and we were preparing to be part of a major new army to push into Germany through southern France. But while we were grouping in France, the Germans invaded the Ardennes, and that battle started, and every American group that was available, Army Division and whatnot, was thrown in to try and stop the Germans, including my division. So we were thrown into the Battle of the Bulge, and I fought in the Ardennes for just about 3 weeks. We were not equipped for cold weather, and many of us got frostbite, myself included. My legs were frostbitten, and I didn't realize how bad they were because they were so numb. But as soon as they put me on a cart, I couldn't walk anymore.
Yeah, right.
And I spent over 3 months in the hospital in Britain recovering from frostbite, and I returned to my outfit just as the war was ending. So I saw some combat, but survived.
Trigonometry to thank.
Yes, yes.
When you came back, you went back to CCNY, and you then went to Yale. So,
Yes, when I came back, I had GI Bill, and I was also a disabled veteran because I had frostbite, and I could afford to go to college and charge tuition. And I applied to go to college at the University of Illinois and Johns Hopkins, but I was turned down by both of them. So I went back to City College and finished there.
So, and then you went to graduate school at Yale.
Yes.
And how did you choose Yale, and how did you like Yale?
I was clearly interested in both genetics and biochemistry, and I talked with the chairman of the department, Benjamin Harrow, which I mentioned before, and he suggested that Beadle and Tatum are combining biochemistry and genetics, and that sounded like just the area for me. And so I applied to Caltech to work with Beadle, and to Yale to work with Ed Tatum. Was accepted at Yale and turned down by Caltech. So I went to Yale. When I showed up at Yale, I learned that Tatum had gone back to Stanford, where he had been previously, and Dave Bonner, who went to Yale with Tatum, stayed on to head the Rossborough group. And so I joined Dave Bonner group.
Right. So and that turned out to be a very good arrangement for you.
It turned out, yes. I mean, looking back, given what happened to Tatum in his research and Bonner in his research, I was very fortunate.
Yeah. Another place that clearly played a prominent role in your career is Cold Spring Harbor. So you were there as a high school student, and then you were there many times thereafter. We talked about 1963, and then you took a summer course there. Did you ever teach there?
No, never taught there. But I give many lectures. I've attended many symposia. As I was growing up, as a member of the Bonner group, we felt that the Cold Spring Harbor Symposia was the ultimate presentation of new developments in genetics, areas related to genetics. And I think we all felt that way. And so we all tried to go to the annual Cold Spring Harbor Symposium.
And at that time, it was by invitation, or anybody could go?
Well, I think at that time probably almost anyone could go from a group that was working in an area that's relevant to the work that was featured at the particular symposium. I'd mentioned that in high school I went there, but in addition, when I was in high school, I wrote and purchased in Cold Spring Harbor a set of Drosophila mutants, which I cultured at home and did genetic crosses at home. My first real genetic experience was in my kitchen at home, growing flies and crossing them for eye color recombination.
So in addition to the chemistry sets and making little explosions at home, you also got to
did a little biology. Yes.
Biology at home, right?
Yes, yes.
And then we had this, you made these pictures of the salivary gland chromosomes.
Well, I
This is high
was
school.
very interested in photography in high school as well, and learned microscopy and learned how to make beautifully stained chromosomes, and that was very exciting to see a salivary chromosome. And so I naturally made pictures of those, and this is a term report from high school, and the title is Cinderella of Geneticist, obviously referring to Drosophila, but the letters actually were cut from photographs of the stained salivary chromosomes. I don't know what this show in the picture, but I've treasured this memento of a very exciting high school experience.
One of the clear messages, at least from both of our careers, it seems to me to be that exposing students to serious science in high school is a very positive thing.
Yeah, I think high school is the time to do it, and I think you just have to give them a little experience, a little laboratory, just show them what's going on in the world, in nature, and I think that a large fraction of them will say, wow. I remember, in fact, how disappointed I was in college initially compared to Bronx High School of Science. The teachers were outstanding at the CCNY as well, but Bronx High School of Science emphasized the future, whereas the City College emphasized these are the facts, this is what you've got to learn. So totally different. But I can't imagine anyone going through the experience that we went through in Bronx High School of Science, not getting out. If you have any sense at all and want to do something spectacular in your life besides just selfishly thinking of yourself only, science can't be beat.
When's the first time you remember seeing tryptophan, or the name tryptophan?
Oh, I'm sure when I learned biochemistry, I learned about tryptophan, but whenever I get asked a question like that, it reminds me of my oral thesis exam, a PhD student, and Sophia Simmons, Joe Fruton's wife, was on my examining committee, and she asked me, how do you synthesize tryptophan chemically? Which is not something I knew much about, so I had to come up with a quick fix. And I came up with a reasonable way to do it, and she didn't know any better, so I got away with it.
So it was one of those theoretical questions,
Yes,
right?
yes. No, it's a coincidence that when I started graduate school, Dave Bonner asked me to determine the intermediate niacin synthesis accumulated by two mutants. He knew I had a biochemical background, and what he assigned me as a project was totally different than what the other graduate students were working on at the time. And so niacin is synthesized from tryptophan, so in learning about niacin biosynthesis, I learned a lot about tryptophan biosynthesis, and I actually studied the relationship in rats initially, and well in neurospora. And I think that in science, in trying to decide what to do next and how to think about things, you naturally fall back on the knowledge that you've gained from what you know. And so I learned a lot about everything that was known about tryptophan niacin synthesis at the time. And so I really knew things that you could do based on what was known about tryptophan metabolism. And so that served as a basis subsequently. At that time period, almost any system that you picked to work on, there were innumerable questions that came up, and answering any one question led to half a dozen other questions, perhaps more interesting because you didn't know the answer. As you know, in our practice of science, once you know the answer, it's uninteresting, it's unimportant. It's only what you can't, anything that you don't know is fascinating.
That's right.
And so it's just, I was following a path.
So the one gene, one enzyme was the big picture that you first wanted to pursue.
Dave Bonner had me going in another direction. Everybody else in his group was working on some gene enzyme system, hoping to get some evidence for one gene, one enzyme hypothesis. In those days, it was not known that DNA was genetic material. We really didn't know that proteins were a linear sequence of amino acids. I mean, but it was very clear from the genetic analysis of people before Beadle and Tatum, but certainly more impressively than Beadle and Tatum's work, that if you isolate mutants that are defective in a particular metabolic function. Those mutants have alterations in a specific set of genes. And if you can narrow the metabolic function to a specific enzymatic reaction, we always find one gene is responsible for that. So I think it's mainly because of just the fact that in working with Neurospora, that you can isolate mutants and correlate with a particular function, that it was natural that since everybody in my group was interested in proving the gene-enzyme relationship, that I would naturally think there's something that I'm working on which would allow me to tackle the gene-enzyme relationship. And in my third year, I switched projects from working on tryptophan, mostly niacin biosynthesis, to picking a model enzyme to try and explore the gene-enzyme relationship.
It's clear that the Beadle and Tatum paper and that whole idea had a big influence. Can you remember any other particular papers that hit you as being really sucking you into a particular line of thought?
Well, there were lots of things going on. Certainly, Josh Lederberg's work on genetic recombination in bacteria was very important. But while I was a graduate student, Dave Bonner made a point of inviting all the prominent leading geneticists in all different areas to come to Yale and to present seminars. So I heard Luria and Delbruck and Tracy Sonneborn made a fantastic impression. All the leaders at that time came and lectured, and like it or not, we've been brainwashed and convinced that this is so exciting. If only we knew the relationship between gene and protein, or gene and enzyme, that would be a tremendous contribution. But we didn't know how to do it. Nobody knew how to do it.
Right. So you heard Luria and Delbruck and Hershey and all of those folks directly. And so in those days, it was, at least for you, it was really hearing them directly rather than reading their papers that,
Yes.
that
Yes.
made the impression.
Yes.
So it was personal.
Although we had journal clubs, which were very important. I remember the first two papers I picked for journal club myself was Chargaff's on ATGC.
Yeah.
What did that mean? And the other one was on the path of carbon and photosynthesis, Calvin and Melvin paper, which fantastic contribution to science, which I still remember describing as in a journal club as a graduate student.
So you finished, or at least you finished your degree in the late 40s.
College 48.
48.
Yes.
And so at
My
that
schooling
time,
was interrupted by the war.
right. We can come back to that. What I was interested in was DNA, because we spoke before that this was before DNA. So in Bonner's place was one of the few places that you could have gone where DNA was on the table already in 1949.
Yeah. Well, 1950s, it was,
Yeah.
I mean, for some reason, nobody took the Avery experiment seriously.
Right.
Okay. But Hershey-Chase experiment was pretty convincing. And of course, by 53, we knew all about DNA, but we didn't know what to do about it. I mean, what amazes me is looking back is that from Watson and Crick, we knew the structure of DNA. You could see what you might be dealing with in molecular terms, but there was no way of sequencing DNA or isolating DNA. You couldn't do anything about it. Eventually, everyone started proposing there must be some code relationship between the elements in DNA and proteins. But that really developed in the mid-50s.
What is the first time that you remember anyone talking about a code? Because it seems to me that before the war, nobody knew anything about codes. After the war, because of the large number of intellectuals who were involved in code breaking in one way or another, code seemed like a natural thing to be thinking about. And I just was curious, when was the first time the idea of a code?
Probably around 1960, in that period. There was a gap in time between the prediction of the structure of DNA by Watson and Crick and when people began to think, okay, what does this mean? And what does it mean in terms of protein synthesis and structure? The first the definitive work showing that proteins are linear sequence of amino acids was also in the early 1950s. So those things happened at the same time, and then it got to the point where you got two linear molecules, somehow they're related, and there were just endless numbers of theoretical articles on how this might be related, and there were a lot of prominent scientists who thought they could predict code without having to do any experiments.
Right.
Yeah. Yeah.
So Ingram's paper on fingerprinting must have made a big impression on you.
Yes, Ingram's paper, definitely, because the first experimental technique to show that you could, in fact, show that the defective human hemoglobin had a single amino acid change.
Right.
And so that all these mutants that geneticists have been studied for decades, in fact, now could be analyzed to ask what is the corresponding effect on the protein product of that particular gene.
So you must have been one of the very first to pick that up as a plan for figuring out about the code.
Yes, I latched on that immediately, but I hate to admit it, but looking back, I feel that progress in science is technology limited, like it or not. Yeah. I like to think that having opportunity of choosing between devoting some effort to developing technology versus devoting that time to solving a problem, that solving the problem is more important. Okay, that's my preference, but nevertheless, I'm willing to confess that the technology really was limiting. I mean, when Wally Gilbert first started DNA sequencing, I sent one of my postdocs. We had spent 2 years sequencing a particular segment of the tryptophan as RNA.
Right.
It was so difficult.
Yeah.
And I sent a postdoc to Wally Gilbert's lab, and he taught this postdoc how to do DNA sequencing. Right. And I think DNA sequencing was pretty slow in those days too. But in 1 month, this one postdoc was able to confirm what 5 scientists had shaky data on based on RNA sequence analysis.
Right. So I think I was in Wally's lab on a similar errand at the same time this was going on. So what fraction of the time in your career would you say you devoted to trying to figure out what was true, and what fraction to actually proving that it was so? How would you divide those? This is the difference between discovery and hypothesis testing.
Well, in my case, that's a very appropriate question, because the 2 major problems we worked on, and one we were pretty sure what we were going to prove, and as time went by, we were more and more confident what the solution was going to be. That was gene-protein colinearity. And the other major problem, which we just stumbled on by accident, was understanding RNA regulation, transcription attenuation, and there we didn't have the foggiest notion what was going on. And every step along the way, it was exploratory. You had to look and come up with some hypotheses and then do experiments, and based on the results of those experiments, then design the next set of experiments. So we knew nothing about what to expect in the second problem. We knew exactly what we expected in the first problem.
But in a sense, a table was set for you in the first problem, in the sense that the idea of genes and enzymes and the relationship preceded, was in a way ancient even by 1950, whereas the attenuation was true discovery, right?
Except the gene-protein colinearity problem, it was redefined. I mean, it's not the situation that the question was defined very early and the answer was provided late because technology wasn't available, but actually the question kept changing. I mean, Beadle and Tatum weren't thinking in molecular terms, in terms of nucleic acids, nucleotide sequences and amino acid sequence, that never even came into their minds. And even the name of the colinearity problem changed. It was one gene, one enzyme, one biochemical reaction hypothesis, and then eventually it became gene-protein colinearity, because we know that a gene was a discrete entity. We knew that a protein was a discrete entity, one with nucleotide sequences, another one unique amino acid sequences. So the definition changed.
Mm-hmm. Were you ever attracted by the hypotheses about, enzymes forming around substrates and the sort of early immunology, George Cohn type of thinking?
Well, I listened to this, but I was not convinced. No, and since so little was known, having the proof became very important. A new hypothesis that sounds reasonable, not good enough.
So we were talking about how the problem changed as time went along, and I'm a little unclear myself as to the exact order in which things occurred, and I wondered about your recollection of where the work by Brenner and by Benzer and Leventhal and all these other people who are also interested in this business, how that interfaced with, on the one hand, the being able to look at amino acid sequences and getting the linearity issues in hand. And I just wondered how you remembered that.
Well, all of it was going on simultaneously, and certainly Seymour Benzer's contribution was, from my perspective, and probably for everybody working with bacteria, and also phage, was very important because he provided convincing evidence that even though we couldn't isolate a gene, we couldn't determine its nucleotide sequence, even though we knew that DNA was genetic material, that in fact you could use genetic recombination to define a genetic map which is a reasonable representation of what linear sequence of base pairs might be in DNA. Mutational alteration with regard to other mutational alterations could be ordered, and the order is not circular, but it's a linear order, and therefore it's a reasonable assumption that this corresponds to a sequence of nucleotides in a gene. And Ingram's work in 1958 certainly showed that mutant hemoglobin had a single amino acid change, and there's every reason to think that corresponding to each mutational change on a genetic map, that you lead somehow to a corresponding amino acid change in the protein. So that's the way the thinking kept changing as the nature of, as we could think of a gene as a linear entity in a recombinational sense, and we could think of a protein as being a linear sequence of amino acids, with one clear example where a mutant had a single amino acid change.
Right. So that was very clear. But Benzer couldn't find the R2 protein for a long time.
Well, Benzer couldn't find this protein. That's right. Other people, Leventhal had a large active group, and Rothman and Alan Garin, and they were using essentially the same approach we were using with, they were using alkaline phosphatase, and this was a workable enzyme. You could isolate mutants, and they were hoping to construct a fine structured genetic map and then use fingerprinting or something like that to determine amino acid changes, and again compare the two. Brenner's contribution really came in later, but he was using a totally different approach. Brenner and the Cambridge group were doing much more fundamental studies looking at the nature of nucleic acids. They were interested in nonsense codons and all sorts of things about the nature of the code. So they were looking at, they came at this from a slightly different angle. Eventually they looked for a system where you could use nonsense mutants to define the endpoint in a gene if in fact you introduced a stop codon in a coding region and that determined the endpoint of the polypeptide. Then the polypeptide up to that point would not be degraded and would survive, and you could just examine its length. So the length of the polypeptide fragment should correlate with a genetic map.
Well, just as Seymour Benzer was unlucky about the R2 protein, Sidney was lucky about the nature of T4, that in fact this degradation is turned off in some way.
That's right, and it was a major protein. 50% of the protein had protein. But I think luck played a major role in all this, and we happened to, so many things happened along the way that was helpful. Like we found a very simple way to make deletions in the TRPA gene so that we could we could order our mutations very easily.
So you were actually able to use the Benzer system, as it were.
Yes, we
in
used
your own.
we all the concept of Benzer in our system, and we used, in fact, the Ingram concept in terms of our protein analysis. And the only the two novel features that our system contributed was the fact that we happened to work with an enzyme that has two protein subunits. Okay? And that was the first enzyme known to have two different polypeptides.
Right.
Now we think it's odd when polypeptide exists free as a polypeptide. They're all an enzyme complex. But anyway, this is an enzyme complex. But most importantly, not only was it an enzyme complex, but each subunit activates the other subunit in the reaction that subunit performs. So A performs one reaction, B performs a different reaction, but A activates B. And so it was obvious to me that if we had mutant A proteins, they might not be able to catalyze their reaction, so isolated as mutants, but that protein might be assayable because it'll combine with a B protein, activate B. And so that turned out to be the case, and so it was very simple for us to work with missense mutants, with mutants with amino acid replacements, to isolate the proteins and then use peptide fingerprinting to find the amino acid change in each mutant protein, and then correlate that with the genetic map. But what took us a long time, say, compared to Sidney Brenner's approach, is that the technology for protein sequencing was very inferior at the time. It took us about 6 years to complete the sequence of the TRPA protein, and probably about 6 or 7 postdocs who worked on that as a major project. But early on, probably 60, 61, we were beginning to identify specific amino acid changes in individual mutants, and it was clear that everything we were seeing was consistent with there being a colinear relationship. That is, mutants which were altered near one another in the gene, they had amino acid changes in the same polypeptide. So we had pretty good convincing evidence, and I guess at Cold Spring Harbor Symposium of 1963, I gave the first real presentation of where we were. It wasn't the complete amino acid sequence, but that was a very exciting symposium.
That was a great
They all were
meeting,
exciting.
Yeah, right.
They all were exciting.
That was a great meeting. Yeah.
And Brenner group solved colinearity using nonsense mutants and measuring the size of the polypeptide fragments at really essentially at the same time. So we both solved the problem at the same time. And also Leventhal's group contributed important information about protein structure. So every group contributed very valuable information relating to the gene-protein colinearity. And so I think in retrospect, I think it's great that a lot of groups were working on the same problem, because had one solved it, I think questions might have been raised, and particularly certainly in our case and Brenner's case, solving it for both nonsense mutants and missense mutants, that that's it. And so everything follows this particular pattern.
You met Sidney Brenner very early in both of your careers.
Yes, yes. Sidney, when I was at Western Reserve Medical School in the faculty and microbiology department, Sidney visited, and at that time he was working on tryptophan biosynthesis and salmonella. He was beginning to do studies trying to identify how many genes and how many different proteins were involved in that particular pathway. So we met at that point.
Right.
And
so,
when we kept in touch over the years.
So there was a lot of communication between
there
you
was
and Sidney.
reasonable, yes. And I was communication with Seymour Benza because of Suppression in the 1955 symposium at Henry Ford Hospital. I summarized all the work, the results of our studies with Neurospora and suppression of Neurospora, and also in those studies with Neurospora, we had a lot of evidence raising questions about what suppression might do. And in fact, that suppression, most important, was suppression restored a missing protein.
Right.
And I still remember after my talk, we discussed, well, what is going on? What is suppression? I mean, if you accept the gene enzyme concept, this doesn't make sense. I mean, if there's another gene that can substitute for your mutant gene, well, why should all mutations affecting a given reaction be altered in initial genes.
Right.
So there's inconsistency. And
So-
we exchanged actually suppressors, and I'm still have copies of those letters, but we both were very disappointed. His were all nonsense suppressors, and mine were all missense suppressors.
Suppressors, right.
And my suppressors did not work on his mutants. His suppressors did not work on my mutants. We just could not understand.
On the other hand, when Alan Campbell and Sidney and Seymour exchanged things, they did find that they suppressed each other.
Yes,
and
yes.
It was all, that was the whole nonsense suppression story.
Yes,
So,
yes. Yeah, so the missense suppression, how we got into suppression, was going on parallel but independent, but we didn't know at the time that there were these two major classes, and so the results were totally different. Why tRNA showed up first was not clear, although now we know it's because tRNA is redundant. So if you have 5 copies of a gene, let's change the anticodon of one and you've got suppression.
Right.
Easy.
Easy, right.
Even with ribosomes, it's not so difficult. You have 6 copies, 7 copies of the ribosomal RNA, but unique copies of the protein. So there are many ways of achieving misreading.
And everybody had the experience, though, that when you had suppression, the protein reappeared, whereas it had disappeared in the mutation.
Yes.
So that was the confusion, because in the case of nonsense mutations, it literally disappears. In the case of missense, it disappears indirectly. Did you ever use antibodies, cross-reacting material? Was that a part of your scheme?
Oh, yes. So we worked with Sig Susskind. We introduced the term CRIM, the cross-reacting material, and we were the first to use antibodies to ask in a mutant, if you examine an extract, the enzyme activity is missing, can you find a protein that's immunologically related to the protein? So I was still trying to find the enzyme to do gene-protein colinearity studies, and I purified tryptophan synthetase from neurospora, and Sig Susskind, just by coincidence, after he was a graduate student with Dave Bonner, but he decided to become an immunologist, and he went to work with A.M. Pappenheimer at NYU, and cooked up this experiment. I gave Sig Susskind my purified enzyme. He made antibody, and then come up to Yale for one weekend. We examined all the mutants, the missense mutants, suppressible mutants, to see which ones would have a protein cross-reacting with the tryptophan synthetase protein. And I still remember most about that as I coded all the extracts. So Sig, he was going to do the immunological test, but I didn't want him to know. And so I put numbers on everything. But it came out, it was consistent.
So when did you decide that E. coli was the way to go as opposed to neurospora?
I was trying to purify the tryptophan synthetase, and it was very difficult. And the neurospora is full of proteases, and there was not much tryptophan synthetase made. Tryptophan is the least common amino acid in proteins, and we didn't know anything about regulation in those days, so didn't know any way to increase the amount of that particular protein. And so I played with E. coli, and rather early in studying tryptophan mutants of E. coli, I discovered that if you grew them on limiting amounts of tryptophan, they produce 50 times as much of the enzyme. So, okay, that was great. And at the same time, there was a genetic analysis. I knew genetic analysis in neurospora very well, and making a fine structured genetic map in neurospora was not going to be easy.
Right.
Okay, but whereas from what the hints from work with P22 in salmonella and so on, it suggested, and also sexual crosses in E. coli, you got the impression that recombination frequency probably was a little bit higher. You could do more in a way of constructing the genetic map by switching it to E. coli. So I just tried playing with E. coli, and I didn't switch E. coli specifically to work on tryptophan synthetase. It just by coincidence it turned out that that enzyme was an ideal subject for analyzing gene protein colinearity.
So much of the early suppression work was done in neurospora. Neurospora was regarded as a favorable.
All the original work leading to the concept that suppressor allows a misreading of an altered template.
Right.
Yes.
So I remember Gross was running around with circular and higher dimensional complementation
Yes.
maps, which is related to this suppression. Were you concerned about that, or did you get involved in that at all?
No, I thought that there was reasonable explanations that we just follow the observation, and that if we saw an enzyme was missing, and that better than that, the enzyme was produced but it was inactive, and we could put in a suppressor and now it restored activity, but the suppressor was specific to the mutant allele, I thought we really were dealing with somehow correcting the amino acid defect.
So did any of your antibodies restore activity by binding to the enzyme?
No.
Because others had found that kind of thing happening. I'm pretty sure that happened with phosphatase sometimes.
Yeah, but
you didn't have that
no,
problem.
No,
Yeah.
no. The RNAs inhibited
Right.
enzyme activity.
So you had a very collegial relationship with all the
Yes.
Colinearity folks.
Yes, we were
Right.
often speakers at the same symposium and
and
brought each other up to date.
it was really a community. Do you think there's a difference between science then and science now in that kind of regard? Because in retrospect, it seems like a very important subject. It was a high-stakes game. In modern days, it would not have been obvious that everybody would just talk to each other and have a good time and bring each other up to date, as you say. Do you think
Well, I think it
has
depends.
changed?
From my impression from scientists I know, it varies. It depends on the individuals and the problem. Some areas of science, they're very great. They're very active in exchanging information, and others, they're very secretive. I think that was true in bacteria. It's a lot. I mean, I can't remember ever going to a symposium where I did not want to tell everyone the latest information from my lab. Not published. This is what's going on right now. This is what we found. So I didn't want to keep anything ever secret.
Right. And you never felt anyone was keeping anything from you secretly
Right.
in those days
Right.
either, right?
Yeah.
That's also my recollection. And
I think the main competition was who was going to get the answer first, but but we're at least talking to one another about it.
Right. Let's go to the other kind of problem, the attenuation issues. The story starts, at least in my recollection, with the relationship of tRNA to the regulation of these enzymes. And you mentioned before that when you grow E. coli and limiting tryptophan, they make a lot more enzyme, and I guess Guarini had done these chemostat experiments with arginine, and it was clear that you could get very big ratios. I guess it was Bruce Ames had showed in salmonella in histidine that the tRNA had to be charged in order for this to happen.
His group was one of the most active groups studying regulation at that time. And all this, of course, Jacob Monod and Pasteur School preceded all of this.
Yeah.
And their work was so persuasive, so impressive, that the prevailing view was that regulation is costly, and that if you can make a simple repressor to shut off transcription initiation, that would be it. That would be sufficient. So one mechanism of regulation, and there were even, remember, arguments examples where scientists found feedback inhibition, and then they thought, well, if you have feedback inhibition, you don't need any other regulation. So we thought that it was so important to each organism, the economy of what it does with its genetic material, that it's not going to devote a lot of genetic material to regulation, just a little. Nevertheless, it can be highly specific. And so Bruce Ames was studying histidine operon regulation. His results were totally different than Jacob Monod's school.
Right.
And so he was finding that it looked like transfer RNA, histidine transfer RNA, not histidine was the regulator. And also the evidence suggested that it may not be the promoter operator that's involved in regulation as well. In our case, Cohn and Jacob had shown that there was a trip repressor gene, and we followed up that, and yes, it was true. There was a repressor protein, binds tryptophan, activated by tryptophan, binds to an Operator is located in the promoter region. That shuts off transcription initiation, gives 80-fold regulation. That could account most of what was going on. So why, I mean, the thought that it might be as important to the organism that two mechanisms of regulation, it's just out of the mind. No one had that possibility. And in fact, there already were some hints that there's something else was going on, and we ignored them because we assumed that the evidence for repression was so convincing that there must be side effects giving quantitative changes in expression. But these side effects are something having some effect on repression, but not indicated a separate mechanism. Okay? And then, but there was one crucial finding by graduate student Ethel Jackson. She was making deletions within the operon looking for the internal promoter. She wasn't looking for regulation at all, and she found that two of her deletions that terminate within the operon near the promoter but not in the promoter increase expression sixfold. Well, what is going on? Okay, and what's even more important, she showed very clearly, if you inactivate the repressor, you still get a sixfold increase. Okay, so no alternative. We had to consider that in this operon, for whatever reason, there were two mechanisms of regulation of the tryptophan operon expression. One repression, but another one involving, ultimately we showed tryptophan tRNA.
In the case of the AIM system with histidine, there is no repressor. Why does tryptophan need both? Whereas histidine is just one, lac operon is just one. What do you think
Well,
of the story?
I think there are alternative regulatory mechanisms. Even with tryptophan biosynthesis, we find in different organisms enormous variation, both qualitative and quantitatively. And in fact, we also find differences in operon organization, some of which are related to regulation and some which are not. So I think it's very difficult to explain the origin of a regulatory process for a given group of genes. Clearly, in tryptophan metabolism is studied extensively in different microorganisms. There are an enormous variety of attenuation mechanisms. So I don't think we can really explain why in histidine, histidine operon has features which allow attenuation to do what repression and attenuation combined do in the trip operon. In the trip operon, tRNA and tryptophan is sensed separately by separate mechanisms.
So one of the things that always struck me as amazing, I always used it in my lectures, is the fact that tryptophan synthetase in E. coli is two proteins, as you said before, but in many other organisms it's only one protein.
Yes.
Including your ospra, as I recall.
That's right. But it's one protein, but it's actually two protein domains that are just fused, they're connected. It's nothing different other than that the ospra, in a sense, is a combination of the two separate polypeptides in E. coli. But many organisms have them fused, and whereas many organisms have them separate.
That didn't worry you when that came out?
No, it didn't work. No. But I'd love to know the explanation. But in fact, if you do an evolutionary screen of genes, there are 7 protein domains necessary for tryptophan formation in all organisms, and all organisms that make tryptophan use the same 7. So they all evolved presumably from a common ancestor. But you find multiple examples of gene fusions to make bifunctional polypeptides, and all different arrangements. So only conclusion you come to, either by chance some of these genes that have to be co-regulated that become fused, and it's simpler to make a bifunctional polypeptide if you have a connector, whereas in other organisms, for whatever reason, it's advantageous to have each one separate, a separate entity. Then you can move one of the genes around. So the complexity is so extreme that I think you can find almost anything to support any argument you want to make from an evolutionary point of view.
So you mentioned a dominant view that came out of the Jacob Monod in the Paris school about regulation, that repression was very intellectually straightforward and very simple, and that's what you needed. Now, of course, very early on, it was clear not only from Ames, but also from the arabinose operon, which was studied by Ellis Engelsberg and his group, that it wasn't universal, and that took a long time to be accepted. How did you feel about all of that?
Well, I became a close friend of Ellis Engelsberg. I took a course at Cold Spring Harbor when I was shifting from the Rossborough to E. coli. I wanted to learn the latest technology in using phage for genetic recombination. I spent the summer at Cold Spring Harbor essentially learning from the Demerits group about genetic recombination. I met Ellis Engelsberg there, and I got to know him quite well, and I became very familiar with his studies. I think Ellis Engelsberg, everything he did led to the conclusion opposite of the conclusion of Jacob-Monod school. Everything he did suggested there was positive regulation, rather than negative regulation. But all of his evidence was strictly genetic. I think Jacob-Monod were wise enough to realize that you have to do something at a biochemical level as well as genetic level to gain complete confidence in the interpretation you've given. Ellis's data was all genetic, and you could argue for each of his observations there was some alternative explanation because he didn't know about something else contributing to it. So it took a while until the evidence became so overwhelming that Engelsberg's work was accepted.
So along the same lines, there came a time when the operator mutants turned out to be what are now called polar nonsense mutants.
Yes.
And the French group was, I think, surprised by this result, even though they had something to do with discovering it. How did that strike you? Because that was also, could have had a sort of a precursor of attenuation. It could have been some kind of attenuation story.
Yeah, well, the whole concept of polarity is an interesting history, and we all were aware of it and wondering what was going on. And you could not isolate mutants in any operon, any multigene operon, without experiencing polarity. They were identified as nonsense mutations with any gene of a multigene operon. If the mutation was closer to the amino terminus of the gene, it's likely to have a polar effect on downstream gene expression. And we looked at the messenger and showed that in fact the messenger also disappeared. And then there was studies with Rho coming into the picture, and it was clear that Rho protein was involved in isolate mutants that are resistant to polarity. And so indirectly this all developed that as a consequence of introducing nonsense mutations, you had premature transcription termination, which is in a sense related to what happens in attenuation, and that turns off production of downstream messenger RNA.
Right. So it always seemed to me peculiar that the Jacob-Monod theory survived the whole polarity episode more or less intact, with the new idea of the promoter in addition to the operator and all that kind of thing. The Paris group had biochemical evidence, but they didn't really have biochemical evidence for the repressor, which would have been the thorough thing. So do you think it has to do with just their priority, or the forceful way in which they wrote and talked about their stuff? Or?
Well, I don't remember the experiment, but I think they did some experiments to distinguish between true operator mutations and polar mutations. And I think suppression was one of the ways out. You could suppress polar mutations, but you could not suppress operator mutations. And so that led them to propose two different classes. Different types of defects, and furthermore, they map at different locations.
Right. No, the mapping in different locations was definitely
a clue.
A clue.
Yes.
So, did you have the same kind of feelings about the collegiality with the regulatory groups? Was it just the same bacterial genetics culture?
Yes, I think we interacted and kept each other informed of what we were finding. And all those areas were hot areas, and so at symposia and annual meetings or whatnot, they were always featured. So, like it or not, we kept up with what we were doing. I mean, there was one point where me and Sidney Brenner and Seymour Benza were at, I think, 5 or 6 consecutive symposia, each talking on the same thing. And we told one another, we heard, but we know what you're going to say.
I wanted to ask you a few things about how your lab worked out. So you mentioned somewhere that at a certain point you got involved in missense suppression as a business, and there was a great deal of work that was being done about trying to understand what the actual basis of missense as opposed to nonsense suppression was. And at a certain point, you decided that your then postdoc would continue with this line of work.
I think nowadays it's very difficult, almost impossible for even an outstanding postdoc to write a grant application on some new field, something he has not worked on, and either get a job or get funding. Whereas when I was first starting, it was quite typical that when you doing a postdoctoral study, that as you were finishing, you would think of something that you had learned from that experience, but you'd work on an independent problem.
Right.
About 1980, that switched, and we're just now, I felt it was absolutely essential when a postdoc left, they would have to continue doing the next series of experiments related to what he did when he was working with me.
Were you ever a postdoc, really?
No.
Is that because of the war, or?
Well, it was very complicated. My last year as a graduate student, Oak Ridge National Laboratory tried to attract David Bonner to move to Oak Ridge. And David picked me and Gabe Lester to go with him. And so we stayed on, assuming that we would move to Oak Ridge. We each made one trip down there. It looked pretty interesting. And we both thought the world of Dave, and we were going to move with him. And that was McCarthy era. And Dave Bonner did not get security clearance, and just by bad coincidence, the Oak Ridge National Laboratory, the biology laboratory, happened to be within a high security zone. If it was outside the fence, there wouldn't have been any problem. So Dave Bonner did not get security clearance. He did eventually, and George Beetle, I think, was one of the major contributors to getting him clearance. Around 1953, both Gabe Lester and I decided to start looking around. And I was at a symposium, I think Johns Hopkins, describing our work on tryptophan metabolism, and some of the people from the microbiology department at Western Reserve Medical School came up to me and asked me if I'd like to move there.
And at that time you were doing tryptophan in your aspirin?
Yes. Right. Although I did do some exploratory studies with E. coli and Bacillus subtilis. And in fact, there's history there that's interesting. Is by coincidence, I obtained the collection of tryptophan mutants of Bacillus subtilis that Burkholder and Giles had produced, showing X-ray mutagenesis, and it was my mutants that John Spization used to show transformation of Bacillus subtilis when we were at Western Reserve. That's curious history. But I was just looking at other possibilities in terms of projects to work on, but I still had in the back of my mind, if I could find a suitable enzyme for Colinearity studies, I would work on that. But when I went to Western Reserve as an assistant professor, I decided to have to pick a well-defined problem to work on. And I knew that the tryptophan biosynthetic pathway was not completely known. And furthermore, there was practically only the enzymology had been done was the last step. And but been hundreds of mutants isolated in dozens of laboratories, and nobody could figure out what was going on. And it turned out, as you might expect, that the reason that they couldn't find the other intermediates is all the other intermediates are phosphorylated. And so you can't feed phosphorylated intermediates. So I worked out the rest of the pathway in Western Reserve. That was the main thing. I worked on suppression, and we still did some work with neurospora, but the main emphasis was working on the rest of the biosynthetic pathway.
So that's more of a parallel with Bruce Ames too, because he was in this exactly the same situation in the histidine pathway.
That's right. He was
phosphorylated intermediates, same business.
Yes.
Yeah.
In fact, not only the pathway is parallel, but a group in Germany has now converted one of the histidine biosynthetic enzymes into a tryptophan biosynthetic enzyme by changing one amino acid.
Really?
That's right. So these are two proteins which are structurally similar.
I wanted to get back to the issue of suppression, because the whole concept of suppression was very important in everyone's thinking about how proteins did their business, and there were all kinds of theories. And did you think of suppression as a principle, or as a technique, or just as a phenomenon, or how were you thinking about it? Later, it became a very formalistic business.
Well, I was taught about it first in a course given by Lewis-Sigler,
Mm-hmm.
and he talked about suppression a bit. And in this course that he gave, each of the students was required to write as common a term paper on some feature of genetics that was unproven. And I picked suppression. I proposed various explanations. And just by coincidence, I got involved in this project because one of the mutants that I was working on and studying tryptophan synthetase of Neurospora, in a control flask, not the experiment, in a control flask, started growing without tryptophan. This is a mutant that required tryptophan for growth. The control flask grew without tryptophan. Why is this mutant growing without tryptophan? So we put it through a genetic course, and lo and behold, it was due to a suppressor mutation. Right. And lo and behold, that suppressor mutation didn't work on every TD mutant, right, but only worked on one of the TD
right.
mutants. Okay, and what's more, it restored enzyme activity. Mm-hmm. So what's going on? Right. I think that sequence of events aroused my interest in suppression, so I stayed with that problem until we showed that suppression did lead to amino acid mistakes, right, in incorporation, and this was all done before we knew tRNA was involved in protein synthesis. Right. And Stu Brody, who was a graduate student working, mm-hmm, on suppression, actually took a course, I think it was at Woods Hole, went, mm-hmm, to a course or some seminar series in which the first studies on tRNA involvement in protein synthesis were discussed. And he just, wow, that explains it. If you have an intermediate that's involved in reading, mm-hmm, information and decoding it by amino acids, that if you mutate that would be suppression. He was the first to show that missense suppression actually led to an amino acid change using the same technology we were using for gene-protein colinearity studies.
So another confusion, of course, was the kind of suppression that's associated with subunits. You certainly observed that kind of suppression as well. Did that confuse you as much as it confused everybody else?
No, we assumed the protein-protein interactions were complex, involved active sites which were complex and not understood. No, that didn't worry. I just assumed that the protein structure and function was so complex that anything was possible. Whereas gene mutations, there's got to be a simple, direct explanation. Okay. Naive, but nevertheless
effective.
Reflected my background.
Yeah, right. One of the things I've always wanted to ask you is, how did W get to be the single letter code for tryptophan?
It was not my choice. So the people who would design it, designating letters for abbreviation, came to tryptophan, one of the last. So they give it tyrosine Y, tryptophan, right, tryptophan W.
I figured you must have a hand.
But the name of tryptophan synthetase,
yeah,
synthetase, desmolase, yeah, also changed 3 times while I was working, yeah, on it, which is pretty annoying.
Oh, well.
The good old days.
So I wanted to go back to the issue of tRNA. We didn't talk a great deal about tRNA, and in particular the central role that tRNA plays not only in attenuation but in colinearity issues and so on. So you had a very long relationship with our colleague Paul Berg, and that started with an experiment. You want to tell us about
we knew that suppression led to mistakes in amino acid incorporation, okay, but what did that mean? So and we had a lot of suppressor genes. What were the genes that were altered that would lead to suppression? And Paul was working on tRNAs at the time. We used to play tennis weekends together, and we discuss our respective research projects with one another frequently. And this was at a time when Paul was well aware of Khorana's ability to use synthetic systems to make RNA-defined sequence. So, and this was also just after we learned about messenger RNA. I mean, in the 1950s, didn't know about messenger RNA. This was a new wrinkle. And so Paul came up with this idea that he could probably have Govind Khorana make us RNAs with a defined sequence, and we could just very simply show that if you have an RNA that's code for arginine, you put in a suppressor that would put in glycine. If you put labeled glycine in vitro, you'd make a polypeptide with labeled glycine. That was the experiment. John Carbon was visiting with Paul at the time. He thought that was a good experiment. Govind Khorana thought that was great, and he had somebody in his lab work on that. So they took two suppressors that we studied most extensively. One was an arginine to glycine, the other one 878, I don't remember what it was. And they made the appropriate templates. We knew enough about the code at the time to know exactly what RNA to make, and sure enough, the extracts of the suppressed mutant would incorporate glycine into the polypeptide. Extracts of the wild type would not. So tRNA was clearly involved.
Right. That always struck me as a great paradigm experiment, because all you had to do was make the E. coli extract, throw it in there, and there you
And
were.
it did
right?
it.
Yeah, it
Yes,
did it, right.
yes. Yeah, we were excited when that worked.
I can imagine. Because you frequently think about experiments like that, and they don't so frequently
No, simple
work.
simple experiments don't come up that often.
That's right.
Right.
Do you have any other favorites? Experiments like that? I mean, not big, long projects, but, you know, where you sit and you say, this is an experiment we ought to do, let's have a go, and it comes out good?
All of them are like that. Everything we worked on, it's amazing where it leads you that you didn't anticipate.
Your work is remarkable for its thoroughness and, you know, finality in many of the areas. If people think about how do we know this, you know, you had a hand in nailing shut the issue. But were there ever times in which you really thought you went wrong, or felt like you were going wrong? I mean, how comfortable were you?
Well, I think any good practice scientist has got to feel insecure with any conclusion he reaches, because there's just so much going on, and it may seem reasonable based on what you know, but you never know everything. And so you have to keep an open mind. So one could never be really sure for anything. That's always been my philosophy.
Do you spend a lot of time thinking about how you should state a result in order that you don't over or understate it? Is that,
Oh, yes, yeah.
Is that a big preoccupation?
Yeah, I think it's important to qualify. Yes, I get annoyed when I review manuscripts when authors have gone overboard, over-interpreting their results of significance.
Do you think science has changed in that regard over your career?
No, I don't really think. I don't think so. I think the review system is pretty much the same that we've had. I think everyone has pretty much the same standards. There have been some changes based on popularity as an issue rather than a scientific merit.
Right, absolutely. Over the years, you've had concerns of a sort of science policy type over favoring one kind of research over another kind of research, especially the issues around is prokaryotic research relevant to whatever, the health of the nation, those kinds of issues. How do you feel about that now?
Well, I still feel that there's a tendency to define long existing problems as being less important and less interesting than something that's a single-step solution, which sounds very convincing, something like horizontal versus vertical science. And I always feel uncomfortable about scientists who devote all their efforts to one very difficult problem, and they continue to make progress. And that doesn't seem to get as much attention as a scientist who discovers some new gene or some new regulator. I think that's unfair. It's not improper recognition of the contribution of the scientist.
But it seems to me that in the long run, finding a new system of regulation, like attenuation, has got to be bring greater rewards than finding yet another instance of repression, for example.
Well, it's hard to measure. You can look back and say, yes, no, for many, many years, considering attenuation, we were told that's such an odd mechanism, maybe a few examples. And then over the years, more and more common, multiple mechanisms. It turns out to be major regulatory mechanisms. RNA is a major regulatory system. Everything changes completely. And that's the progress of science.
At the time that you wrote your editorial in Cell, which is 1991, about the concern about that prokaryotic research was being neglected, it seems to me that a restorative development has been the whole business of genomics, and the extent to which it's now become absolutely clear that all the proteins are derived one from the other. So how do you feel about the whole genomic developments?
Well, I think the knowledge that it's generating is very valuable. You could spend, in my generation, spent their whole career studying one group of genes, and now you can begin to ask questions about the whole genome. And I think that's very valuable information, and it's great. Anytime I think there's a new technology, a new set of information, I want to know that, if it's possible to obtain that. But it's not going to tell you the mechanism, and ultimately, after we understand all this mechanism, we really understand how evolution, how did these things get this way? There's got to be some basis for the end result that we see in nature now. Okay? And so this information is very valuable from an evolutionary point of view, but it's not, you can speculate as to what's going on, but I think it's going to require additional levels of experimental technology to exploit genomic information to work out networks and regulatory components of networks, localization, and message turnover, and lots of variables.
So when is the first time that you remember thinking about the whole issue of evolution, and how much has it been an influence in your thinking about biology?
As soon as we knew something about repression and attenuation, we wanted to know there are other organisms use the same mechanism. Attenuation, we sequenced the leader regions for a whole bunch of different bacteria and showed significant differences, some of which we could explain, some of which we couldn't. And in terms of protein, the same question came up. As soon as we knew the amino acid sequence of the E. coli TRPA protein, we wanted to know, well, what happens if you do the amino acid sequence of other proteins? How do they vary? Why are residues conserved? And so it seems to me that, from my perspective, the type of science that I do, once you understand mechanisms, you're obligated to next think about evolution. How did it get that way? What is the significance of the differences that you now see?
You also mentioned several times the relationship between biochemistry and genetics, which I think is the hallmark of molecular biology.
With biochemical genetics.
Biochemical genetics, right. Yeah.
Before.
Is molecular biology a discipline? How do you think about that, or is it just biochemistry and genetics applied to a set of problems?
Well, I think it's probably a realization that what might be arbitrarily defined as separate disciplines in science have to be connected. You can't discuss genetic material without discussing the biochemistry of the products, and you can't you can't consider biochemistry without knowing something about how genes, the structure of the genes and how they're regulated and how they got the way they are. The trend now is for more and more areas of science to become interrelated and to feed one another to give a broader interpretation of observations than was possible when they were separate.
You're a professor emeritus now at Stanford, and you taught all these years. What did you like teaching?
I like teaching.
Yeah.
I enjoyed it. It was the pleasure you get from the reaction of students or postdocs or graduate students is more pleasure than you get giving pleasure to yourself by doing something. Okay, it's the second-hand enjoyment is I find more rewarding. Teaching is fun, but it's very demanding, and I still cannot understand how anyone teaching high school or junior high school can teach every hour for many consecutive hours. I mean, when I gave a course with 3 1-hour lectures a week, that's practically all I could do during that week is plan exactly what I wanted to say in those lectures.
So did you ever consider doing anything else except becoming a professional scientist?
Actually, yes, when I was in college, I worked for one summer as an assistant in a company that does organic chemistry. They're making polymers and so on. So I got to experience some chemistry firsthand, and I was not impressed. No, but actually, in my background was such that I really didn't have any understanding of what an academic career could be like. Nobody in my family experienced anything that would give positive feedback from that experience. And I remember talking to David Bonner, who was my advisor in graduate school, and telling him I thought that I'd go to graduate school or go to work for some company after that. And he used to laugh. He said, "You're not going to end up. You're going to be professor in some school doing research and teaching." Me? No, I don't want to teach. I want to do something, constructive in the way of doing what somebody wants to have made in an organic chemistry laboratory. Okay, totally different way of looking at it, but fortunately I was educated differently.
So I take it that if you had to do it again, you would do the same thing.
I would choose the same thing. Yeah, I can't imagine any pursuit in life that's going to be more enjoyable than what we do.
Well, I think that I wanted to thank you for doing the interview, and in particular, thank you for all the good example that I think you've set for all of us on how you actually do this profession, which is maybe not so simple all the time. Thank you.
And thank you for asking me the tough question you did. Thank you.